welcome. Good morning. I'm going to be talking about active site prediction, more specifically about functional site prediction, but we're going to focus on active site prediction. I spent a few moments looking at, writing down some notes. Let's see if I can get them. So here, functional sites in proteins, many different types of functional sites. They range from enzyme active sites to specificity determinants to allosteric sites, cofactor binding. There's many different types of functional sites. And biologists are very interested in this for a number of reasons, at the top of which is experimental investigation is expensive and it takes a lot of time. And it really helps to winnow down the number of sites they have to test by having predictions. If you can tell them of the 300 residues in their protein, there are 3 that look like they're catalytic sites from one source of information or prediction protocol to another, they can quickly test those, and then that makes their life a lot easier. Otherwise, they have to test all 300, and that's a lot more expensive. And then active sites are found typically in clefts or pockets, and if you do computationally assisted drug design, computer-assisted drug design, you can target pockets that have what look like active sites for different types of drugs. Benchmarking methods are very difficult, and so I will show you why that is. And the key information, I've left off one type of information. I'm going to see if anyone can see which one I left off. So there's 3 different types of basic information. One is sequence and structure conservation. So sequence conservation means when you build a multiple sequence alignment, and you all know what a multiple sequence alignment is, right? Is anyone here who wants me to remind them what a multiple sequence alignment is? Nobody's going to admit to that. Okay, so let me just remind you what that is. Multiple sequence alignment is a matrix, and if you have a column and it has a perfectly conserved position, but there's variable positions everywhere else, that's called sequence conservation. It's family-wide. You might have other positions where the residues that are conserved are only conserved within a subfamily, but not across the entire family. That would be called subfamily-specific residue conservation. All right, subfamily-specific or family-wide. Family-wide conservation is more typical of enzyme active sites. Subfamily-specific conservation is more typical of specificity determinants or ligand binding residues in families where there are many homologous groups. Solved structures, 3D structures of high quality, which means very few gaps in the structures and good resolution. Solved 3D structures are very useful because they tell you where the clefts are. There are software tools that you can run on a 3D structure that will tell you you've got a cleft here, you've got a cleft here, and so on. And those cleft detection tools are very important for predicting enzyme active sites because active sites are typically found in the clefts. And certain residues are much more likely to be catalytic than others. Does anyone know what residues are more likely to be catalytic than others? Anyone? We'll start off with charged residues. So D, E, K, and R, and also histidine, serine, asparagine, which are not charged, but they're polar. So polar and charged residues are much more likely to be catalytic than hydrophobic residues. So the more you go into bioinformatics, you're going to need to know the amino acids, what they're like, what types of roles they typically play. All right, so what did I leave off as a key source of information in predicting catalytic residues? Anyone have an idea? It's related to what I was talking about last time I was here. Anyone? Is it too early in the morning? You didn't get your cappuccino yet. It's annotation transfer. Okay, so now if you have a let's say this residue. Let's say this residue is found in an enzyme, and someone in a lab somewhere tested that residue and said it's catalytic, and then published their work in a paper, and that publication was read by someone at a database that curates catalytic residues, and this could be SwissProt as an example, or the catalytic site atlas of the EBI. And they would enter that the D at this position in this protein, let's just call that protein X, was found to be catalytic. Then all the other proteins that have a D in the same position in a multiple sequence alignment or in a pairwise alignment can be predicted to be catalytic. It's a form of annotation transfer. You're transferring annotations at a site basis. So this is one of the key things that happens in bioinformatics. In the context of active site prediction, it happens at the context of individual sites. You can say, if position 221 in protein P is catalytic, and if 235 in protein Q is homologous to P221, so we're going to just say that this is actually not a good annotation or way of representing it, just to this residue. Then position 235 in Q is catalytic. So that's the prediction. You want to see that? So this is a form of annotation transfer at a position. You have this position, which is catalytic. This position, which is homologous. We have different types of edges in a graph that represents all these. So if these two are homologous sites, then you can transfer the annotation of catalytic to this residue. Now we see this in automatic function prediction in BLAST, for instance, where you take an entire protein and you run BLAST and you find a match, and protein 1, let's call this protein P and Q again. The entire function for the protein, for protein P, can be transferred to the entire function for protein Q if you say they're homologous. Now there are issues with this approach, but it's the same basic idea. You're transferring an annotation based on homology. Here we're transferring an annotation based on homology, but it's one site. So it might be this site. Right? So site-specific annotation transfer and a global whole protein-based annotation transfer. Got that? Does that make sense? It's a pretty simple idea, but it's important that you understand it. If it's not really clear, come up to me in the break and write down your questions that you want to ask me during the break. Come up, I'll put them on the board, and then when we come back in the second half, I'll answer those questions before we move on. Okay, so this is just to get you started. And now we're going to go into the slides. And a lot of these ideas, or most of them, or all of them really, are in the slides. All right, so many different types of functional sites. We've been talking about catalytic residues. The reason I focus on catalytic residues is that in the bioinformatics community, there is a benchmark database called the Catalytic Site Atlas, which is a very good benchmark. It's not complete, but it's been used widely, and there are hundreds of residues that are known to be catalytic in the literature. And now we get to a critical issue, which I'm going to jump into. One of the issues in machine learning so really you have two basic types: supervised learning. Does anyone know what the other basic type is? Supervised learning and unsupervised. Good. In supervised learning, you need a benchmark data set for training your method. So you need training data. That's why it's called supervised. This wasn't within the scope of what I was supposed to talk about, but it's relevant to later on in this lecture. If you've heard this before, have they heard this before about training data and model overfitting and things like that?
Not extensively.
Not extensively. Okay, but you've heard about training data. When you have training data, what happens is you have a set of parameters. Let's call this a bag of parameters. for your model. And you want to fit the parameters to the training data. So this training data is going to be used to modify the parameters. Got it? Now, if you fit your parameters of your statistical model to your training data, does that tell you how well your statistical model will perform on data that's not in the training data? Let's make this kind of clear. If the training data were restricted to eukaryotes, this is just an example. Eukaryotes include all animals, fungi, plants, all sorts of weird little organisms like protozoa. They're all eukaryotes. If you have a model that you've trained entirely on eukaryotes, will it work when you try to apply it to bacterial families? Maybe or maybe not. It might be really working very well on the species that you trained it on, but not on the ones that you haven't trained it on. So one of the critical things in building a statistical model is you take your training data, and first of all, you want to understand the limitations of your training data. If your training data are restricted to a certain, let's say, lineage, taxonomic lineage, or certain types of enzymes if you're doing active site prediction, you have to be aware that it may not work very well on other types of data. That's the first caveat. But typically, you're going to take your training data, and you're going to do k-fold cross-validation. Has anyone here heard of k-fold cross-validation? There are some nods. Okay. So you do k-fold cross-validation, and I can talk about that another time later if you want me to talk more about it. But it's a way of dividing your data set into a bunch of pieces. And what happens is you pull out one piece, train on the rest, and then test your method on that one piece, which is to say, you make the predictions on the piece that you withheld. And then you record your performance. Where did you make mistakes? What did you get right? And then you put that back in the data set and take out another piece and set that one out and train on the rest and report your performance on that piece. And you repeat until everything that's in your data set has been used for testing. And then you give your average performance over the entire withheld data set. That's called k-fold cross-validation. It's an important technique for benchmarking. When you're reading papers, if what you read says that people took the entire data set, tuned their method on the data set, and then report their performance on that data set that they've been training their method on, that's cheating. People don't always realize they're cheating, but it is cheating. And it's cheating because the performance on that data set does not tell you how well their methods can perform on something that statistical model hasn't seen before. So the aim of benchmarking is to give a feeling for the generalizability of a statistical model to unseen data. That's a really important point. Now, if your benchmark data set has flaws, if it doesn't list things that do have the characteristic you're looking for, or it lists them as not having that characteristic, or some of the things that it says have the characteristic don't have the characteristic, then you're basically training your statistical model on noisy data. That is an unfortunate reality of all bioinformatics tasks. So I'm going to show you that as well. All right, so we're going to focus on catalytic site prediction, primarily because we have a lot of data for it. So what are catalytic sites or enzyme active sites? I haven't actually confirmed that this slide is still available, but when I put together these slides at the very beginning, I got this image from that website at the bottom. Essentially, they're found in enzymes, so catalytic sites are in enzymes. There's typically a small number, 3 or 4, that are strictly conserved for the family and that are essential for the enzymatic function. Other residues may be very important for maintaining the shape of the cleft, or for binding a cofactor, or for binding a ligand. So they have different roles. But that cleft itself here, that shape of that cleft may be different based on what substrate is bound. So you get the subfamily determining residues. but the whole region around here tends to be very conserved structurally. So you can use structure comparison to say, in regions where there's an enzyme active site, in a family that has diverse functions, the catalytic residues will be conserved, often perfectly conserved, and their 3D structure is going to be perfectly conserved in the enzyme as a whole. So you can use structural conservation as well as sequence conservation to detect those. Here's an example of a protein that's not an enzyme. It's a sarcomology 2 protein. It's a domain that's found in many different proteins, and it's involved in different cancers. The sarcomology 2 domain has only a couple of residues that are perfectly conserved. And here I'm showing them. This is from my PhD thesis, which was a long time ago. The red and the purple, perfectly conserved. And these light blue residues are the ligand binding residues. And they tend to be conserved within subfamilies, but they differ from one subfamily to the next. So when I was doing my PhD, I was really interested in finding subfamily-specific conservation patterns. And so the method I came up with, which ended up being used in the annotation of the human genome, was really designed initially for finding these subfamily determining residues. The Catalytic Site Atlas is one of the key databases that we used in my lab for benchmarking our methods for active site prediction. But it's actually changed. So here's some references down here. These are really fundamentally important papers if you're interested in this field that you want to read. And this one here, Analysis of Catalytic Residues into Enzyme Active Sites, first author Bartlett in 2002, is what essentially gave us this idea for the statistical model that we wanted to put together to recognize active sites. But they have an update. So the Catalytic Site Atlas is now the MCSA, and this is the front page. So you can go to it and you can search in this using a Swiss prot identifier or a P2B identifier or the enzymatic function. You can just write it out in text like asparagine synthetase. So the CSA dataset started off with hand-curated entries. So they're going into the literature. They have a small number of people on their team who are reading the papers and entering them into a database. And then, in addition, they found homologs using SciBlast, and they used an e-value cutoff that was very small, which you can see there. I don't recall how many iterations of SciBlast, but I would expect it's probably 3, because that's typical. It's very common to do 3 iterations of SciBlast. And then they annotated those residues as potentially catalytic based on homology, and they required that the residues agreed. So it would not be enough to have a residue at that position that was different from the one that was catalytic. So they required if it was a D in the original entry, it had to be a D in the homolog. And we've already talked about annotation transfer. So this is from that paper. What are the characteristics of enzyme active sites or catalytic residues? Not all biologists or biochemists will agree with this definition. They will quibble with certain aspects of the definition. But this is the definition that the authors of the Catalytic Site Atlas used. It has to be directly involved in the catalytic mechanism, or it can alter the pK of a residue, or it can stabilize the transition state, or they can activate the substrate in some way. So these 4 different ways of being called an enzyme active site. Doesn't have to be all of them, just one of them is enough. And that's from Porter et al. In 2004. So the information from 3D structure, active sites are most often in pockets or clefts. Typically it's in the largest pocket. If it's not the largest pocket, it's in the second largest pocket. They're typically solvent accessible. So what does that mean? When you have a protein, hydrophobic residues tend to go toward the center and they don't want to be in contact with water. The surface residues are solvent accessible, at least somewhat. And for a residue to be catalytic, it has to be somewhat solvent accessible. It doesn't have to be completely solvent accessible. The side chain doesn't have to be sticking all the way out. It could be largely buried. But some part of it has to be solvent accessible. And there's a very interesting point about the B factors, which is essentially how much a residue can wiggle. And that's in the Bartlett review That's very interesting, so I recommend you read the paper and read about B factors. The secondary structure, they're typically on loop regions. They're not on helices. Most often, they're on loop regions. That doesn't mean they can't be on helices or beta strand, but more often they're going to be on a loop that's projecting into a cleft. So when we build a statistical model, we're going to have those features: secondary structure, solvent accessibility in a cleft, right? We want to have those features in a statistical model for recognizing enzyme active sites. Evolutionary conservation is something for which you actually need homologs. So for these guys, all you need is a PDB structure that you can interpret. If you have a PDB structure that's deposited in the PDB, you can download that PDB structure. You can use tools to find clefts. There's software tools that will say it's clefts at these positions. You can use tools to say which positions are beta strand, which are helices, which are loops. And that will tell you about these sites. But when we get to evolutionary conservation, that means across homologs. So now we need a way to bring in homologs and align them. So if two proteins have the same function, I've already used much of this chalk. When proteins evolve from a common ancestor, they can diverge in function. So let's say this is group A and this is group B, and that there's a duplication event at this point in time. If you go very far out, if these guys are fairly closely related, even if they're two different subfamilies or paralogous, they may still have the same catalytic residues. But now let's say you go far out, let's say these are eukaryotic, and now we get into some really divergent in function. And now you don't see the same residue conserved in this group. So now instead of a D, you've got an E. So it could be that you have a different enzymatic function showing up in this family. The further out you go, residues that are catalytic based on the CSA definition may no longer be catalytic. And that you would really wonder if the residues now start to be more variable. So instead of even being conserved within a subfamily, they're variable. So here, conserved D, now we have E, Q, and N. These are all polar, and they're similar to each other. They're also similar to D, but they're not showing conservation within this group. So that's one of the types of patterns you can see. So even in a family of related enzymes, the far enough out you go, the types of sequence conservation that you might see will change. I've talked about this earlier, so now you're just seeing this is for your notes, because all these slides, you can download them from the website. Chothia and Blesk, I don't know if you've seen this paper. It's a fantastic paper about structural conservation across homologous enzymes. And they showed that the regions around the active sites are highly conserved across evolution, but other parts tend to vary. So there's a conserved core structure. This concept of a conserved core structure also comes up in protein structure prediction. The Eisenberg from David Eisenberg's lab, they have a method called 3D cluster analysis, which makes use of this type of structural and sequence conservation around active site residues. And then a method from my group, the discern algorithm, which I'll be describing, also does that. So I'll be presenting in this lecture the Eisenberg lab method as well as the discern algorithm and other methods too. So the types of features that a statistical model can exploit include the catalytic propensity or the catalycity of different residues, conservation patterns, information from 3D structure, and combining all of that. All right, so now let's look at a few different methods. One of them is called evolutionary trace. Has anyone here heard of or used the evolutionary trace algorithm? Evolutionary trace is historically important. If you're interested in active site prediction or specificity determinants, it is a method that you can make use of because there's a web server for it. Olivier Leach-Charge came up with that algorithm around the same time that I was working on my method, and we got to be friends, and so our ideas were very similar, but we went at the task with very different approaches. 3D cluster analysis came out of UCLA, the University of California, Los Angeles, David Eisenberg's lab. Olivier Leach-Charge was originally at University of California, San Francisco, and so this was a University of California proliferation of methods in this area. I was at UC Santa Cruz. So all of us across the University of California are working on this, but there are many other people across the world working on it. FR-PRED, I'm not actually going to be talking about that much because it takes more time than I have for this, but this is one of the other important methods that you want to know about. Syfy was a method I developed as a graduate student. Then Intrepid went from my lab at Berkeley, and then Discern also at Berkeley. And then these other methods that you might want to know about. These are also machine learning methods, Goodrich et al. And Yoon et al., which also used machine learning techniques, statistical modeling techniques, and information from evolutionary conservation and structure. So first thing I want to tell you about is,
what is the input of all the methods? Do you need like a protein ID, or you need information?
All right, so the question is, what is the input for the methods? So if you go to, I just spied a piece of chalk so I can actually write some more on the board. If you go to the Evolutionary Trace website, you have two different ways of interacting with it. One is, I believe, you put in a protein identifier, like a PDB identifier. But I think it's also possible to paste in a protein sequence in FASTA format.
Yeah, because the idea is that you put in information about the protein or an enzyme that's unknown, right? So you can predict parts of the protein.
You would hope, but the evolutionary trace algorithm may, of course, have trained a model based on the protein that you've actually submitted. You may not realize it. It may have already seen your protein. It may know a lot about the protein that you want to know about. He may have brought in information from literature in order to train his method. So for you, it's unknown. For them, it's already known. Okay. You've heard of entropy, Shannon entropy. It's a measure of how much uncertainty there is at a position in a molecule. So if you take let's look at this position in the tree, just this subtree. This position with a completely conserved D is going to have very low entropy. If you look at pi log pi, if the probability of a residue being present is 1, log of 1 is 0. So you can have 0 entropy. Entropy is maximized when you have a uniform distribution over all the possible types of characters that it can have. So entropy is high when you have high variability, and it's very low when you have low variability. So entropy is a natural way of assessing which positions may be important functionally, because if evolution is requiring them to be conserved, it's requiring them to be conserved either for functional reasons or for structural reasons. So if you recall the scorpion toxin superfamily that I showed you that has insect and plant defenses as part of that superfamily, there were a small number of disulfide bridges that are formed by cysteines that were perfectly conserved in that family. Those cysteines are important for maintaining the structure. It holds those small molecules together. But some positions are conserved for, let's say, enzymatic sites, so the different types of conservation. So entropy is a natural way of getting at which residues are important. But I wanted to point out to you that entropy, if, for instance, now we look at, if we were just looking at this subtree, and we have 3 E's, a Q, and an N, and now I'm going to take an adjacent position and pretend that it has, let's say it's this 3 I's, so it has the same types of, and W and K. So in this case, we have the same number of characters that are being seen. So the entropy formulation is going to give you the same result for this column as for this column. But in fact, this column is at least conserving a type of amino acid, which are polar amino acids, polar in charge. These are all across the board. So isoleucine, tryptophan, and lysine are very different types of amino acids. So entropy is not a perfect way of representing the types of structural variability you might see in a protein, but it's a statistical way that is widely used. And there are different types of entropies: relative entropy, which is also known as Kullback-Leibler divergence, and we'll be looking at other forms that are also very useful. Then there's relative entropy. In relative entropy, you can look at the similarities between two distributions. So, for instance, if you're trying to align two groups of sequences to each other, let's say you have a group of sequences aligned in this subtree, a group of sequences aligned in this subtree, and you have aligned them separately because you're using some type of bottom-up alignment protocol. So you're aligning things that are similar to each other first before you're trying to align two dissimilar groups. That's a very common technique in agglomerative clustering, or in a way of building a phylogenetic tree where you're actually using the sequence alignment, or a way of getting an alignment where you're using the phylogenetic tree. You can represent the amino acids at a position using a probability distribution of the amino acids. And you can represent this group using a probability distribution. And then you can use the relative entropy between those two probability distributions to say what alignment makes most sense. And that technique is one that I've used in my lab. Many people use it because it's a powerful technique. But then you get to the question of how do you form the probability distributions? Do you just use the raw frequencies of the amino acids that you see in that alignment, or do you round them out with similar amino acids using a Blossom 62 substitution matrix as you would in BLAST and PSI-BLAST, or using Dirichlet mixer densities, or some other technique? I'm leaving these slides in so you can read them at your leisure. A lot of these mathematical concepts require just slowing down and thinking about them. So I encourage you, just when you have time, just work through some examples with relative entropy. You might want to code it up and just see what happens when you try to actually use relative entropy between distributions. Then we get Jensen-Shannon divergence. So Jensen-Shannon divergence is a symmetrized form of relative entropy. So if you look back here to relative entropy, you'll notice that it's not symmetric. You have two distributions, P and here F. Often we're going to use P and Q to represent the distributions. But it's not symmetric in P and Q, or P and F in this case. But Jensen-Shannon divergence is a symmetrized form, and that gives it certain very important characteristics. So it's smoothed, which means there are no ways for it to blow up, because it takes the distance to a midpoint, and it's symmetrized. So this is a very popular technique, and it's one that we've used and been able to convince ourselves that it's more effective than most of the other techniques. All right, so now historically, let's go back to evolutionary trace. So this was in 1996. This is at UCSF. Olivier Leescharge came up with this idea. It's a beautifully written paper. It doesn't require any mathematical or machine learning or statistical training. It's a lot of the biological intuition. So even if you don't love heavy math papers, and I know only a few people in the world who love heavy math papers, and they're all mathematicians, this is a really nice paper. And I'm just going to walk you through the algorithm. The algorithm is naive. Well, it's not naive in a biological sense. It's actually really deeply informed in the biological sense. But from a computer science, computational perspective or mathematical perspective, it's really simple. And that's what essentially gives it its power, because it embodies the biological intuition. So what they did is they took a family of proteins that they're interested in. They constructed a multiple sequence alignment. From the multiple sequence alignment, they constructed a phylogenetic tree. And now they're orienting the tree so that the root is on the left. And you'll note that I virtually always draw my trees the same way. So if you're looking at trees in the literature, you might find that the trees are drawn in this kind of funny way in a curve where you have like things coming out all over the place. But if you're trying to look at a multiple sequence alignment, that makes it impossible. So when you're looking at a multiple sequence alignment ordered by a tree, put the tree on the left-hand side, and then each leaf on the tree, which represents a sequence, and has the sequence going out from it. You have your multiple sequence alignment on the side. As I draw here, it makes it easy to interpret. And then they took vertical slices of the tree. So starting at the root of the tree, and I'm going to give you another residue. Let's just pretend that all of the proteins in this tree have a conserved arginine at that position. So they start at the root of the tree, and they say what residues are conserved across the whole family. And then this residue is the only one that satisfies that criteria in this toy example. And then they take a slice, the first slice that they can find, and that breaks up the tree into two subtrees. Now they say, now what residues are conserved across the whole family? Right now, from this example, nothing else is conserved, so I'm going to make up one. Let's make this an N. And let's make it change to an S here. So at the first, this one is going to be found at the first cut, and that'll be found at the second cut. Got that? And then they're going to continue pushing this cut of the tree from the root out towards the leaves. And at every point, it's going to induce a partition of the sequences into distinct subgroups. You guys know your set theory? You know about partitions? A partition is a discrete cutting of a group into different non-overlapping groups. So every vertical slice induces a partition. That partition, you can then examine every subfamily and say, is it conserved within that subfamily? And if it is, you would mark it as being conserved at that cut. And they continued all the way down to the leaves. Then the next thing they do is they go back and they look at the 3D structure. And they had some software that would highlight the positions in the tree, positions in the multiple sequence alignment on the 3D structure that were conserved at that cut. So for instance, and this is really, unfortunately what I've given you is impossible to see. You'd have to read the paper. But early on at the A cut, you have only a few number of residues that are perfectly conserved. And they would show up, let's say, here. And as you turn the structure, you'll see some residues are highlighting at one point, at one cut, and not at other points. By the time you get all the way out to the leaves, all the residues in the structure will be highlighted. So then they're looking for an optimal point in the cut of the tree, such that most of the residues that are highlighted are only showing up on one face of the molecule. So it's very manual. It requires a human to be rotating the structure and looking at it. It also assumes that your tree is basically correct, and it assumes that your multiple sequence alignment is basically correct. But that's the basic idea that they had. So from doing that, they found that they had really good prediction accuracy. Compared to what was available at that point in time, it was essentially the best available web server when they put together a web server. So now Olivier Lischarge went off to another university and formed his own lab at Baylor College of Medicine in Texas and developed this web server. And you can submit sequences. I haven't checked that it's still online. I assume it's still online because it's been online for decades at this point, and I think he's got reliable funding, but I can't confirm that it's still available. So you can put a PDB identifier, that's a PDB identifier. PDB identifiers have 4 alphanumeric characters and followed sometimes by a chain. And this would be a uniprot accession. So you can put a PDB identifier or a uniprot accession according to your question, and then click submit, and it will give you results. You can also download the software, I think. All right, so that's one very simple method, and here's Syfy stands for subfamily classification in phylogenomics. So the input is a multiple sequence alignment, and you can see it's very easy to find these perfectly conserved positions. The output is a phylogenetic tree with a cut of the tree into subfamilies. From that cut, you can then find positions that are conserved within subfamilies in addition to the residues that are conserved across the whole family. And then you can build a set of subfamily hidden Markov models. So this idea used information theory and some agglomerative clustering and Dirichlet mixture densities to round out the amino acid distributions. It's actually a fairly simple approach, and it works surprisingly well. So here's essentially the most interesting thing about it really is the encoding cost. So how do you decide where to cut the tree? So we've talked a little bit about agglomerative clustering. Remember agglomerative clustering? It's a bottom up. Everything is a leaf in the tree. You find the two closest, join them, come up with another way of representing that group that could be a profile. And then you join the next two closest until you have a rooted tree. You're repeating until you have a rooted tree. So that's agglomerative clustering. You can use this in many different application areas. In this case, we used profiles using Dirichlet mixture densities and sequence weighting. Profile scoring using relative entropy. And then that's the distance function, so we symmetrized it. And then to cut the tree, we used a minimum description length under an encoding cost for the set of alignments that were derived from that particular cut in the tree. And that encoding cost is here. So n is the number of sequences, s is the number of subfamilies determined by that particular cut. So this is saying for every sequence you want to encode the label that says it belongs to this subfamily or that subfamily. So you need log base 2 bits if you have S subfamilies. If you have one subfamily, you don't need to encode the label. Log of 1 is 0. If you have two subfamilies, you need 1 bit. It's either 0 or 1. If you have 4 subfamilies, you're going to need 2 bits, right? So for every sequence, you need to have some way of saying it belongs to this subfamily, that subfamily. In addition, you need to encode the individual subfamily alignments. So here we have 2 subfamilies, this group and this group. And if we had to encode them separately, we could say, okay, it's easy to encode a conserved R, a conserved D, a conserved N. Those residue distributions are well known and they're easy. You need only a few bits to encode them. If you have a complex group of amino acids like this one here, 3 I's, a W, and a K, that's a hard probability distribution to encode because it's very rarely seen. So you need many more bits to encode it. So you're encoding for every column, C, and for every subfamily, the log base 2 of the probability of the amino acids or the probability distribution seen at that column under the Dirichlet mixture density prior, represented by theta. I think this is probably in the detail a little bit much for you, especially at this hour of the morning, and especially if you haven't had probability statistics and Bayesian tools and information theory. But what it tells you is some columns are easy to encode. You've seen them a lot. Conserved amino acids, similar amino acids are easy to encode. This column with 3 E's, a Q, and an N would be cheaper to encode than this column because this column is seen a lot. This column, which a lot of you can't see, and maybe now you can. So E, Q, and N are all polar amino acids. They're found in many of the same environments. So it'd be cheap to encode these under Dirichlet mixture density. I'd have to explain how Dirichlet mixture densities are derived, but they're derived from multiple sequence alignments. So they know what amino acid combinations are likely. This column would be very expensive to encode because it's very rarely seen, that kind of collection of amino acids. All right, so then what happens is you've got this encoding cost function. You're going in this agglomerative clustering, and clustering everything is in a distinct subfamily, so it's expensive to encode everything independently. And as you continue, you start clustering things that are similar together, your encoding cost drops. and you find a minimum point, and then you go back up again when you start bringing things together that are dissimilar. So you find the minimum encoding cost, and that defines the cut of the tree into subtrees. And here's an example of how that works. So this is from a phylogenetic tree that we built at Sjolander Genomics for the functional annotation of the human genome. And these blue diamonds represent subfamilies that were found by Syfy. And each one of these represents a distinct type of protein, so different somatostatin receptors. This guy is a singleton. There's only one sequence in that subfamily. Allodostatin receptors, and here's an orphan. These are gallinan receptors. Here are opioid receptors. So you're seeing that the cut of the tree to subtrees conserves specific functions within the subtrees. And that was the intent, but it was surprising that it worked so well. And then you can actually compare phylogenetic tree topologies with Syfy, and you find that there's a lot of consistency. The well-defined groups functionally are found by phylogenetic tree methods as well as by Syfy. Now we looked at SH2 domains, and in SH2 domains, there's one position, this arginine, which is perfectly conserved, except in this case, this is a sponge, where that protein was a fragment, so we only saw the last part of the protein. But that arginine and that histidine are perfectly conserved across the entire family, and that's an example of family-defining residues that anyone could see just from looking at the multiple sequence alignment. But all the other subfamilies had some type of variability. So this one allows a G here, this one allows a C in that group, otherwise it's an S. High variability in this column, including polar and non-polar amino acids. This one also has some high variability. That one has a lot of variability. That one has a good amount of variability, including non-polar amino acids. And so you see each position, there's like some real variability. This one is a little bit peculiar, IV, compared to all these others. So you have this kind of variability of conservation patterns. But the positions that were most important for phosphopeptide specificity, that's this one, which is highly conserved within each subfamily. It's only one position where there's a single residue substitution, isoleucine and valine, and they're very similar amino acids. So you can use this technique to divide up a family into subfamilies and find positions that are really important functionally by looking at these different types of conservation patterns. Now we get to, I'm just going to go a few more minutes and then we'll take a break for whatever you get, your 5 or 10 minutes, and then we'll come back. 3D cluster analysis identifies interfaces and functional residues, and what they did is they constructed a multiple sequence alignment. They did not use a phylogenetic tree, so they really wanted to see what they could get just from structural conservation. And then for every position in the molecule, they looked at positions in the alignment that were within a certain distance in angstroms from that residue in the molecule. And they constructed a subset. They extracted the corresponding columns, and then they looked at the conservation similarity in the conservation within those positions only. And they could score every residue in the molecule according to how well conserved the structural environment was. So that's the basic idea. And in fact, it worked decently well, but it's not competitive with methods that also use evolution or statistical models that use other types of information like in a cleft or on a loop or the secondary structure. But it was a very good idea to essentially show how much information you could get out of it. Now I'm going to stop here because the next half starts getting a lot more complicated.